The talk that I'm going to present today as part of the twosome that me and Dr. Chang are going to cover is an area that we are really excited about because this is a new tool that we want to be able to deploy in our approach and efforts to be able to bring life-changing nutrition that will impact the pets and, of course, the people who love us well. Because keep in mind, many of these tools are just as equally applicable for human subjects or human applications as well. So there'll be two parts to the talk, as we alluded to. The first part, I'm going to attempt to answer the question why. Why are we getting into this journey? And then I'm hoping, and you'll see a much more elegant description of the how piece that Dr. Chang is going to be presenting. So in a sense, I'm the cover band, and he's going to be the main show that you're here to watch. So let me begin by the first slide that really talks about two milestones. Now, mathematical modeling is not new. It's been done for over decades. But 2 points of note that I like to highlight here was the Nobel Prize in Chemistry in 2013. And this is given to this team, Dr. Martin Karplus, Michael Levitt, and Ariel Warshel. And what they demonstrated over 40 years of research was an elegant way to be able to predict how proteins behave in biological systems digitally, in silico. And that's fascinating because it opens up a lot of different opportunities for people to explore this. For instance, you can start thinking about how drugs or drug-like compounds would interact with a protein, helping open up opportunities for new drug therapies. It can help us understand how enzymes would interact with the ligands. It can even help us understand, for example, how does photosynthesis occur in the plant cell, which is kind of the fundamental basis of how there is life on Earth. So amazing opportunities to be able to explore. And those tools have been already deployed quite extensively in the pharma industry as they start looking for new solutions for treating various diseases. Let's move forward a decade later in 2024. And there were Nobel Prizes awarded to 3 individuals, but there were actually 2 sets of work. Demis and John Jumper, their role was with a company that is associated with Google, and what they showed was elegant ways to be able to predict protein structure using just the sequence information. But what's interesting, at least in my mind, is the work that Dr. David Baker did at the Howard Hughes Medical Institute in Seattle was trying to come up with novel ways of creating unique proteins that had amazing functionality, all digitally, and then of course you can start synthesizing those using various molecular biology tools. Now this really opens up again an amazing array of opportunities for us to explore. For example, can we start looking at devising proteins that can transfer a drug-like compound or active compound to various parts of the cell, like molecular motors, or you want to call it as cellular DoorDash. You can actually deliver things directly to some part of the cell. You can start looking at ways to be able to create nanomaterials using proteins that have, again, unique properties. So it really, again, opens up a whole array of opportunities for us to be exploring, and I'm sure many of us in the room are aware of this, and this is an exciting time to be able to do research in this space. And I mentioned earlier, the future is here, and it is for us to be able to embrace it. And you might say, hey, that's a long way out, but just think of things that have changed over the few decades that we have experienced. For example, how many of you remember the Motorola Razr phones that they were the pride and joy of many of us? And in many cases, they were actually given freely by these companies. But today, they're no longer here, and we have these amazing smartphones. We don't talk as much on these phones, but we do everything else: shop, look at videos, send emails, send texts. And of note is that every time I have to buy a new set of these phones for my family, I've got to take a second mortgage, right? So things have changed quite dramatically over time. But the point I want to make here is that these evolutions and revolutions are going to come up much quicker than we can imagine. Now, you could say that's great, that's all amazing, but what does that mean for us? So let's start looking at how does it impact what we do as a nutrition company. So there are two things I want to propose or consider as we think about that. The first one is these are tools that can really enable us to be able to do quick analysis of how protein-protein interactions can help drive decisions on what ingredients we could look at for potential diets that we may be able to propose. Just think of the research pipelines that we all run, all of us involved in this process. You have the early phase, which Dr. Johnny Lee elegantly demonstrated in the previous lecture, systems biology, omics, identifying pathways that are disrupted in disease. The same can also be applied to a situation where you want to be able to enhance a particular pathway in health to maintain health. So application both in the disease scenario as well as maintaining health over time. And if you identify particular enzymes, particular molecules that we want to impact, we go back to our toolbox. These are usually nutritional ingredients, and figure out ways to see how some of those can interact and change the trajectory, change the function of some of these receptors, proteins, to be able to deliver the health benefit. Now, what these tools that we are going to talk about, and Dr. Jang will give you some examples, elegantly helps us do these things so much faster. You can literally screen through thousands of compounds in a matter of days. And what that does us is to be able to get a list of molecules that are likely to have, or ingredients that are likely to have significant benefit quicker, so we can start getting down to the business of actually doing this in a clinical trial to show the benefit. Now, when you talk about all this digital stuff, all this modeling stuff, one thing that always keeps coming, and that's a very important and relevant question. In all these cases, you're predicting the probability of success. But at the end of the day, all of us here are really interested in saying, does the diet make a difference for your patient, for your pet, for me? So at the end of the day, all these things are great tools, but they will live and die in the clinical trials that you'll be running. And that's the second important point I want to make, because if you look at the research portfolio, the amount of money we spend on others, in our case, we want to be able to put those dollars, more accurately Swiss francs, on the later part of these studies, because that's where you have to be able to run robust, highly powered clinical trial studies or nutritional studies to be able to show the benefit of a diet that you're exploring. So if we're able to do the earlier part of research in identifying potential targets quicker, you're likely to be able to have a much greater chance of getting successful outcomes that'll eventually land in terms of solutions that you can use to treat your patients much more effectively. So that's, I think, that the key idea is it shortens the span of getting some of these researches done. It helps us put money where you really need to be able to drive these clinical trials. And hopefully there'll be money left over for us to do more studies in addressing more challenging situations. I think I covered most of those in the rambling that I went through, but the key point again is AI and computational models can really transform how we look at nutrition research. And more importantly, these simulations or sophisticated simulating platforms can help us optimize clinical trials, which are ultimately the bread and butter of what we do. Again, you've seen the computational approaches that were talked about again in Dr. Johnny's talk and talks over yesterday as well. Great tools for us to be able to look at small molecule metabolism, combine large data sets to be able to see patterns that are otherwise impossible to visualize just looking at the data by itself, and then drive unique insights that can lead to novel solutions. There will be three kinds of modeling platforms that Dr. Jang will talk about. I'll just tee them up. Docking stimulation, where you're trying to understand how smaller molecules would interact with larger proteins. Molecular dynamics, to see how these proteins behave in various biological systems. And traditional machine learnings that will eventually lead to things like biomarkers, etc. Now, as you can imagine, we are very early in this stage, and you could wonder why talk about it now, because we are super excited about what this can deliver for us, and we want to be able to share it with all of you so that we can get some feedback as well, because this is a team effort. We are not doing research on our own. We're working together to be able to advance the science so that we can benefit the pets that we love and the people as well. So there's going to be some early discovery work that Dr. Chang will cover. It's too early for us to be able to talk specifics in terms of what molecules, but the key idea for us is to be able to give you a flavor for what we are engaged in, what's coming down the pike. And I'm really hoping that in the next few years, we may be able to stand here to be able to talk about specific examples of how a nutritional solution evolved from some of the work that was initiated at this stage. With that, I will turn the podium over to Dr. Chang.
Thank you, Dr. Satyaraj. Great introduction. My name is Chang Woon Jang. I'm a data scientist and molecular modeler at Nestle Purina. Well, what I'm going to talk about today is actually the more details about actually the specific examples of the in silico modeling Dr. Satyaraj introduced in the earlier talk. Actually, I'm going to show you about actually the three specific case studies using in silico modeling and molecular dynamic simulation. The in silico modeling typically include widely used actually the pharmaceutical industries for drug discovery. This includes actually the in silico protein ligand docking simulation, and also to identify the screening large set of the actually the natural drugs or natural compounds using computational simulation. So molecular dynamic simulation is a little bit more robust and then computationally expensive and then slower than protein ligand docking simulations because it needs actually the more parameters, mathematical parameters. But molecular dynamic simulation is very popular nowadays in various industries and then academia to predict complex biological system properties or engineering materials. So we know about actually the data-driven modeling, machine learning model. Nowadays, actually, they're very popular as well to predict actually the unseen properties from the very large set of the data. So let's move on to actually the case studies. So the first question is how we can utilize actually the in silico modeling to find a better nutrition solution at Nestle Purina. So it is obviously very effective tools to screen actually the large food libraries in terms of the cost and time-wise. So imagine that if you test actually the 70,000 or more than 1 million compounds in the laboratory, it is literally technically impossible, right? But computer simulation can help this way to screen in a very fast way. So in my experience, actually 70,000 compounds can be screened actually easily in a day or two. So also this ligand docking simulation to easily identify potential hit through actually the ligand and protein interactions between two target proteins. And also it is very helpful understanding actually the interaction mechanisms between the actually the ligand and docking ligand and target proteins can guide actually the finding effective compounds by feasibility assessment. So let me talk a little bit about actually the ligand docking simulation process. So this is very straightforward. You can just create actually the structures of your interest. So first of all, you can create actually the protein model using, as Dr. Satyaraj described in the earlier talk, you can use a homology modeling or AI modeling tools just to use by protein sequence information. And then you can identify binding site, active binding site. This is very important because the binding site will activate actually the protein structures for protein function by binding, ligand binding. So, and then you can create it actually, your interest of the ligands, as many as you can, then you can put it in the active binding site to calculate the binding affinity or binding energy as an indicator of the binding affinity. So this is a pretty simple concept to understand about ligand binding affinity. So it is a competitive way to binding to the active binding site for known reference ligand. I can show you later. So, but how do you believe it is reliable or not? So we can just compare the crystal structures with a predictive model. So you can see here is one of the ligand, known reference ligand. And then we use the ligand docking simulation to compare and then superimpose between the crystal structures and then predictive model. It's pretty match well between the known crystal structure obtained from electron microscopy and our predictive binding mode. So now we believe that it is very reliable because we predicted the crystal structures. Then the next step is we can just calculate all the binding energy to and then compare the reference binding energy to pick up what is the best option, best effective compounds to inhibit or activate the protein function. So here is the first showed the model 1, model 2, model 3 actually showed all compounds 1 showed the best binding affinity. That means the compounds 1 is the potential compounds to inhibit or activate. But ligand docking simulation doesn't reveal the compound's behavior, but another way we can look at the interactions between the ligand and target proteins. So here left figure showed the known the agonist reference ligand. So the protein activity is well known by the ligand binding. So you can you can search the many literatures what is the interactions between the ligand and the proteins in the binding domain. So here threonine and then tyrosine is key residues to activate the protein structures, but just only the 7.4 kilocalorie per mole binding free energy shows. So our new compound showed the better binding affinity with the lower binding energy. So potentially you can see this new compounds interact with different residues. However, it has the binding affinity. So it competes the known reference ligands. So it's more stronger bind into the binding domain. So it pushed away reference compounds we know. So in this case, we can assume that this could be potential antagonist to inhibit the protein structures function. So now let's move on to the molecular dynamic simulation, which is more robust computational technique. But this molecular dynamic simulation can enable us to understand physical motion of the complex biological system or any engineering system at the atomistic scale. So, and then it is easy to, it is very useful way to predict actually the macroscopic properties or behavior of our interest complex systems, such as protein stability or ligand behavior. Because the ligand, when ligand binds to actually the active binding domain, if the ligand is agonist, the protein structure conformation will change rapidly. However, if the ligand is antagonist or inhibited, actually the structures is more stable and over the time. So this is one of the example of this biological system. So we can obtain actually the complex ligand binding protein structures from the ligand docking simulations. Then you can put it in actually the water. So this mimic actually our certain biological system. And then you can put it in the actual parameters, physical parameters. Then actually we see that actually what is actually the protein structure conformational change. So one is actually the properties, actually the interactions between actually the ligand and proteins. So here you can see the top right actually showed actually the hydrogen bond, number of the hydrogen bonding over time. So you can see the number of the hydrogen bondings they remain very consistent. That means actually that this ligand doesn't activate actually the protein structures. So could be, we can assume that actually this is the antagonist behavior to inhibit actually the protein function. And another one is actually that you can put any environment conditions such as temperature, pressure, you can change whatever you want actually. So in the bottom of actually the right, so the protein structures change it by temperature. So you can see this is the beta-lactoglobulin with triglyceride actually the bind to the beta-lactoglobulin. So when you change actually the temperature, we find actually the first until actually the 70 Celsius degree, the RMSD, root mean square displacement of the backbone chain of the protein is very stable. But suddenly, those conformational change actually by RMSD results rapidly increased. That means actually the 100 Celsius degree will change the temperature where the actual beta-lactoglobulin completely denatured. So this is a pretty good approach to understand actually the biological system using MD molecular dynamics simulations. Another one is the quick topic about actually the machine learning approach. Machine learning approach, we used actually, we realized actually the machine learning and statistics to identify key biomarkers from omics data sets, such as the proteomics and metabolomics. So machine learning model, as you know, can discover actually the key biomarkers and then predict in this study actually the biological age, and then predict actually the disease progression of the heart disease of the canine called MMVD, mixed mitral valve disease. And then also we can utilize actually the statistical analysis to identify actually the statistical significance between the groups. So the first example of the machine learning study using proteomics data sets. So this is a simple background is about actually the medium chain triglyceride associated the metabolic change in young and senior dogs. So we can determine actually the biological age based on actually the proteomics information. So protein profiles. Usually proteomics and then omics data has high dimensional data, which consists of the thousand different proteins from blood samples. So we can detect actually the key proteins as we use actually the feature importance algorithm. And then first, this plot actually showed actually the R-square values the 0.68, which means actually the 68% of the dependent variable is explained by actually the all features of the protein profile. This is pretty good actually, the result actually, because the very large dimensional data has actually the noise. So our goal is actually how to reduce actually the noise, and then pick up actually the key biomarkers from the very large set of the protein profile. Then we used actually the feature importance algorithm to select actually the 24 proteins, and then retrained actually the model, and then showed actually the better performance to predict actually biological age. So ML model is very useful, can identify key biomarkers, in this case is proteins, and predict pretty good in way biological age. And then the second example of actually the machine learning study we performed at Nestle Purina is canine heart disease, which is actually the enlargement of the mitral valve. So 79% of the dog with heart disease have MMBD, myxomatous mitral valve disease. The mechanism causing MMBD are not completely understood. However, the studies by Purina scientists indicate that the specific blend of the nutrient slow down actually the progression of the MMBD and help actually the improve the heart function in those specifically with when we find actually the disease progression at stage 1 or stage 2. So stage 2 or stage 3, I'm sorry. So stage 1 represent actually the health conditions, and then stage 2 and stage 3 is actually you can detect actually the progression of the MMBD, and then stage 4 indicate actually the failure of the heart. So this is a question, how ML model detect actually the early stages of the progression of the MMBD using serum metabolomics. So our target is actually the detect actually very early stage of the B1 instead of the B2. So I first visualized actually the sample distributions on the machine learning spaces. So you can see control and B1 is slightly overlapped because there is a similarity between actually the similarity of metabolites between two groups. However, when you see actually the B2 and heart failure stages, the groups actually moved slightly toward actually the right. That means we can assume that actually the metabolite change over actually the progression of the disease. So then I plotted actually the relative, mean relative Euclidean distance between the groups. The stage control and the stage 2 early stage is very close to each other. However, control and heart failure is the very far away each other. That means actually the heart failure metabolite actually drastically changed. So as I said in the previous slide, our target is actually detecting the early signs of the disease progression of our companion canine animals. So we adopted actually the support vector machine to separate actually the control and then B1 early stages. So in this case, we used actually the nonlinear support vector machine to separate actually the two groups. It showed actually the 85% accuracy. It's pretty good classification performance. So the key message is here, machine learning model can predict actually any type of the disease progression possibly and detect the early sign of the disease. So we can make actually the nutrition intervention to prevent actually the disease progression and the help actually to improve their health companion animals. And then in silico modeling is not just actually focused on actually the certain studies and researches. So in silico modeling can support all 5 fundamental our Nestle Research Institute. I already supported actually the health science, food science, agricultural science. The packaging material is actually the engineering field. So molecular dynamic simulation, here is the bottom of the plot showed actually the simulation can follow the experimental observations. Even though, because we are only model actually a small portion of the engineering material, does not mimic heterogeneous structures of the real samples. So summarize of my talk today is in silico modeling approaches has tremendously increased in research area nowadays, including in silico modeling, ligand docking simulation, and the machine learning model. So it is very cost and then time effective for studies and researches, and it is easy to perform. So it allows actually the high throughput screening, helps select nutritional compounds very, very quickly. And then also it enables actually the deep understanding of the molecular events, and then you can easily visualize actually what happening at the atomistic level using your computation. And then we can build obviously unique complementary knowledges through the computational modeling. So I believe the increasing demand for computation-driven research is more and more in the future. I would like to acknowledge actually my Nestle Research St. Louis team for their support and then contribution and dedication to my research work. Thank you very much for your attention.